I'm Charles Chung. I'm at Wayne State University, as was said, and I know that this topic's a little bit different than many of the topics we've had in the series. So hopefully this will be a kind of a unique introduction into how I view relaxation and what we're trying to study. Before I get too far into this, I want to first highlight that a lot of the work that I'm going to be doing was performed by undergraduate students. So on the top here, there's 4 undergraduate students who've recently graduated and moving on to medical training, clinical training. I currently have 2 graduate students and another undergraduate student, along with technical assistants in the lab. And really, what I'll say is most of the experimental work has really been done by these students. So what I want to talk to you about today is really how I got into this and what does it mean for the future. So I'm going to talk briefly about kind of my background and why we've been pursuing this, what it means when I say mechanical control of relaxation, and then what I think the mechanisms are, which is myosin and maybe other mechanisms. So we'll step through this and we'll start with the big picture view. The big picture kind of, I'm in Detroit, so the highway view, the interstate view of the physiology of dysfunction before we get into kind of the streets of titin and myosin and some of these other ideas. So of course, we hopefully all know the Wigger's diagram with isovolemic relaxation and then filling. And of course, we know that slow diastolic relaxation is bad in that when we increase our heart rate, the primary adaptation is actually to reduce our filling time. And so that becomes a serious issue in our long-term health. Delayed relaxation in and of itself is considered a marker for HFpEF, and it is present in nearly all heart failure and many other comorbidities. So it is fairly important in our ability to be active in our lives. So I'm going to talk about relaxation, but I got into relaxation by starting with diastolic filling. So I want to bring you there because it'll relate back into how I think about this. In my predoctoral work at Washington University, we thought about the heart as basically a spring. And when we're in diastole and that spring is stretching, then that really well characterizes the filling of the heart. So this is a transmitral Doppler E-wave. So for example, we would be sampling the velocity of blood coming into the heart. And as many of you know, we have early rapid filling and late atrial filling. And using a simple physical model, including inertia, resistive losses, and stiffness, we're able to really well characterize this. What that gives us is an opportunity to look at the physical properties of the heart in terms of resistance or damping and stiffness. But when we look at relaxation, the historical theme of relaxation has always been kind of this trimodal view, which is in order for the heart to relax, you get the cytosolic calcium concentration to reduce, you reduce thin filament activation, and then myosin cycling causes the forces to decline. That is typically characterized by an exponential decay. And being that I have a physics background, we started looking into this. And as many of you know, if you take the pressure and plot it in the phase plane where you have dP/dt versus P, relaxation is this straight line as we can calculate from these rates. Now, this is an exponential equation, and we can rewrite this equation. And really, this is basically telling us that tau is a damping term. Now, this is good, and this is what almost everyone uses when they report relaxation rates. So if you are doing pressure volume in a mouse or rat or even a human, this is how we would typically measure the isovolemic relaxation. But it was noted by, I think it was Suga's group in 1995, that there are definitely hearts that have a curved shape in their relaxation, a curved phase plane. And so they proposed this logistic equation. So now we have got a choice. We can pick a mono-exponential or a logistic equation. And so when we look at the variety of hearts that are available to us, we can see we can either choose a straight line or a curved line, but there are certain ones where the choice between a straight line and a curved line really doesn't feel appropriate. And these are fits that I did on real clinical data when I was a pre-doc. So the question for me when I was looking at this was what is it that, or is there a way we can create an equation that fits all of these in one go? And the way I went about that was to first think about whether or not relaxation was just decay. And as I said, when we were looking at our filling, we have stiffness in addition to the decay. So what happens if we, instead of looking at filling, look at relaxation as inertia, resistance, and stiffness? That changes our equation a little bit. And we've got now two parameters related to relaxation. And I don't want to go too deep into that equation because it's not as important for what we're talking about. But what I do want to point out here is on the top is that same graph I showed you before, but on the bottom, this is the fit to all of the different relaxation components. So you can see that one equation, including elastic stiffness on top of the relaxation, actually really well characterizes all of them. So what we're finding is that by balancing the stiffness and the relaxation components, you could actually modify how the heart is relaxing. And so this is what drove me into kind of the molecular biology, because I was really very focused on this very big picture, high level physiology. So because I was interested in this elastic term, as Jonathan mentioned, I went to do a postdoc with Hank Kranzier. And so I went kind of deeply into the physiology of Titan. Now, I don't want to go too deep into the background of Titan, but hopefully most of you realize that Titan is this elastic spring that spans the sarcomere, or the half sarcomere, I guess. We have isoforms, and those isoforms modify the cardiac stiffness, not just in the muscle level, but at the whole heart level. And we've shown this many times. The other thing that you may or may not realize is that Titan's very important in helping the heart, or helping a sarcomere, re-lengthen after it contracts. This is an experiment that was done in Doug Sawyer's group, I believe, by Michael Helms, where he took a skin myocyte and had it contract and then washed out the calcium. And then you can see a very rapid re-lengthening of this muscle. When they degraded the titin in the muscle, they again caused contraction, but you can see the rate of relaxation of that muscle is severely inhibited. So titin, I thought, would be really important in helping the heart go from that contracted state to the relaxed state. Well, after many years and looking at many of the knockout models that I worked on, that Hank has had in the past, we found that there's basically no consistent change in isotonic relaxation in these mice. Hank's also published at some point with the Stell Lab in Germany that these mutant mouse models where they modify titin really doesn't change the relaxation of a myofibril. So this is, again, this is a single strip of myofibers, myofibrils. And there's actually two colors here. You may or may not be able to see that. There's a black and a gray. And one of them is the wild type and one of them mutant. And that's pretty clear that there's not much difference. So I gotta say that this was kind of a dark time for me. I was hoping that titin would solve all of my problems, but it didn't. So I went back into the big picture physiology and try to think about relaxation. Now, hopefully I haven't lost too many of you, and I'm going to go back into a little bit of relaxation itself. And of course, many of us know, as I talked about before, contraction and relaxation. Contraction is calcium coming into the muscle, to the cytosol, copper to binding sites, and thin filament activation and myosin activation. And that's what's causing forces. We of course know the ATP cycle, where ATP binds to myosin, causing relaxation, and then it gets hydrolyzed to cause force, and then we get a power stroke as PI and ADP are released. And so this is what's really driving the force generation. And when we have more ATP, we drive more relaxation, of course. So the traditional biochemical concept of trimodal relaxation is, again, calcium leaves the cytosol, closing binding sites and unbinding myosin. So we can look at that a little bit within an intact muscle. So a lot of my experiments right now are working on intact cardiac trabecula. This one happens to be a papillary muscle, and there's some differences on that. I'm not going to get into that today, but you can see we can pace an intact cardiac muscle, and we can plot its force and length. So when it's excited, we can see a contraction and we can see a relaxation. And again, that relaxation is primarily driven by calcium removal, thin filament deactivation, and myosin detachment. But some of you who are interested in cardiac relaxation know of the work of Brutzer starting in the 70s and many others who said that there's actually a mechanical intervention that we can do to accelerate relaxation. And this theory is out there, and you'll see the hints of this in even the trials justifying interventions in the the RAS system for HFpEF and antihypertensive treatments for HFpEF. And that's that afterload, or the arterial blood pressure, can modify relaxation, and that reducing afterload might accelerate relaxation. So one of the things that we tried, and a lot of this was based on the data acquisition system that Ken Campbell developed, and he really drove a lot of this part of the technical aspect, in that we can take a muscle and then instead of doing an isometric twitch, we can load clamp it and pretend we have an afterload. So we can pretend we have an afterload and then let the muscle relax, and later on, I'm not showing it here, but we can stretch the muscle as if it's filling in diastole. Well, we can do that at any different afterload we want. And I'm not going to show you the quantification, but many of you can see here that really these relaxation rates are all basically the same. And so it didn't work. And I got to say that I was at this point with Ken, and he kind of looked at me and was like, what are we doing? Why are you doing this? And of course, I had to go back and read the literature, which is frustrating for a postdoc, but super important. And I had to go back and reread not just the reviews that were written, but also the work from the 70s to find out that, in fact, the primary work that this is based on is shown in an example on the right. So we recreated these graphs as if they would be as they would be shown in Brutzer's work. And the one thing that he stated in these studies is that he actually performed these studies with relaxation loading. What that means is that the muscle was being stretched to maintain this force. And so you can see here the relaxation rates of these muscles becomes very different when we start looking at these different afterloads if we have this relaxation loading. So we reiterated in our study that the stretch was actually necessary to modify the relaxation rate. Now, of course, some of you who are keen on your physiology are thinking, well, that's pretty strange because in a normal heart, we have contraction ejection, where we change the volume or length. We have relaxation and filling. If we use relaxation loading in its maximum extent, we have contraction, ejection, refilling of the heart before the aortic valve would close, and then relaxation. So there would be no cardiac output on here. Well, this of course can't happen in the real heart. But what turns out is that even from 1921, or who knows, perhaps earlier, it was noticed that there is the cessation of contraction before isovolumic relaxation begins, and actually there's a slight twist in the muscle. So this is the original Wiggert diagram from 1921. And you note he points out this protodiastolic phase before the isovolumic, or what he calls the isometric relaxation. More recently, we of course have imaging data. This example here on the right is simply MRI strain data, so principal strains along the long axis or the circumferential axis. You can see here isovolumic contraction, there's not a whole lot happening. And then you get ejection, and we have the strain dropping dramatically, and then we have a stretch of the muscle again before the aortic valve closes, before isovolumic relaxation begins. And then, of course, when the mitral valve opens, you get dramatic changes in strain again, where the heart begins to fill. So this is actually physiologic in that there are a lot of hearts that will stretch before the end of systole. But what's also interesting is that if you look at the time to peak strain, in patients, for example, with hypertrophic cardiomyopathy or hypertension, again, these are patients that would regularly have impaired relaxation or regularly reported to have impaired relaxation. They don't stretch before the heart relaxes. They stretch afterwards. So we looked at this a little bit further, and we started to do moderate stretches, kind of mimicking the physiology. We've actually done the full range of stretches, and what we found was that there's a relationship not about how much it stretches or the afterload, but it's actually based on how quickly this is stretching before the heart relaxes. This relationship, this slope, is what I call mechanical control of relaxation. So if we can stretch the muscle faster, it will relax faster. We tested this in mouse, rat, and human trabecula. Ken Campbell has an excellent resource of human tissues and an excellent collaboration down there. And when I was there with him, I was able to collect some human trabecula and show this. And you can see all of these species have a relationship between relaxation and strain rate. So this is where my current consideration of what is relaxation and what is diastolic function. It's that physiologic stretch is necessary and sufficient to modify relaxation, and we want to look at what this means in terms of how we can modify cardiac function. So as I mentioned earlier, there are some hearts that just don't stretch or don't stretch before it relaxes. And so that's probably one form of diastolic dysfunction. But there's probably other forms of diastolic dysfunction, and that might be related to this, this relationship. So what I'm interested right now is what can we do to modify this? Is this relationship attenuated in disease states? Are there treatments that we can accelerate this? One thing I didn't put in here is that when we reduce afterload, for example, we are actually modifying the strain rate and not the not other relationships. So in this original paper, we talk about that. So for example, if you're treating a heart and you're making physiologic modifications, you might not be modifying the relationship or the afterload, but you might actually be shifting where you are on this graph. And regardless, if we're down here and we want to move up, it would be better for us to move not only up further, but up further and faster. So we're looking at these mechanisms that might modify this. And I want to stay in the big picture physiology for a few more minutes, in that we're doing some experiments in the last few years related to preload. So this is work that was done by an undergraduate, and we've just submitted for publication. We did these experiments where we can contract the muscle, we can do these load clamps, we can do these various stretches. We can also calculate the mechanical control of relaxation from this data. And then we reduced the preload. When we reduced the preload, we can repeat all of the experiments. But what happened was that the slope went up. So reducing preload improved mechanical control of relaxation. This is repeatable. We took a muscle and went back and forth between the low and high preloads. And as we reduce preload, it goes up, increase back and forth. We've also done a couple of muscles where we do intermediate preloads. And again, we maintain this relationship. So mechanical control of relaxation is preload dependent. And there's a clinical implication of this, which is that reducing preload or volume would actually improve relaxation. And I would suggest that this might be an explanation of why some of the clinical trials or clinical studies that do, for example, reduce afterload by using diuretics might actually improve relaxation. But now we want to know about the molecular mechanisms. I kind of shotgunned this. I said, well, what happens if we reduce preload? I didn't think it would actually change, but it did. But in order to move into the molecular side, I want to have a little bit more of a direction rather than shotgunning these studies. So in order to do that, we've been doing some computational modeling. We're using the myosin system, which was developed by Ken Campbell. And what it did in our previous study was really help us identify that it was likely that these stretches are detaching across bridges very quickly, and more quickly than they'd be normally detaching, and that's what's accelerating relaxation. So this is a nice method for us to take data and see what happens. So we did that. There's some limitations to the model. The solid lines here that I'm plotting are the model fits, and the dotted lines are data. And so you can see here our relaxation is actually kind of weird. We're not precisely getting strains either, so there's some issues with that. But what you can see is that we have some slow relaxation and then some stretches that had fast relaxation. And that's consistent with both muscles. So we've been spending several weeks, and I had a really great physics undergraduate student who's now going to, I think he's going to UCSF for med school. So I'm going to lose his ability to do computational models, but he went through and did several models for me. And I don't want to take too much time on these because this gets a little bit complicated. But what I want to say is that we did have a couple of models which did increase the slope, increase mechanical control of relaxation. But it turns out this one here that my mouse is closest to suggested that the thin filaments were more active at shorter sarcomere lengths. So let me get into that for a second. So this is just some model scaling that we did recently. So we had the model, as I showed you before, we have peak force. This here is the force that we would have the peak isometric force for the short length, and this here would be the load clamp force. This gray line here is the mechanical control of relaxation that we calculated with the L0 condition. And we took all of those same mathematical parameters and stuck them in our model, and we got this dotted lines part right here. Now, the data that I've seen published says that at lower sarcomere lengths, a muscle, the thin filament is less likely to be activated. So in that case, we would actually predict that mechanical control of relaxation will actually go down, not up. We've done this for a number of other parameters. For example, basically the troponin off rate and cooperativity. And overall, what this is telling us is that the thin filament activation would have to increase to make the mechanical control of relaxation increase. So we're probably not looking at any of the thin filament parameters in this. What we are probably looking at are thick filament parameters. So the parameters here go from attaching myosin. So we've got detached myosin to attached. We've got a power stroke. We've got detachment. And then we've got over here a strain rate dependence. And what I'll point out here is that all of these can modify relaxation. We don't actually expect the attachment rate to change too much as far as the data that I've seen. We do expect the power stroke to get shorter though at low length. And that could drive us into this realm which we've discussed before, which is to really move up mechanical control of relaxation. And we can do that without dramatically increasing the force of the system. So this is just kind of a playing with model idea to say, well, if we do what happens physiologically, we might actually really have a sort of reasonable modification. We have a detachment rate, and then we have here strain sensitivity. I don't have a lot of data on this, but this is something that's of huge interest to me. It's basically, is it easier or harder to pull off myosin head the farther away you get from its baseline position? But the thing I want to point out to you here is that there are physiologically reasonable transitions that, or kinetics that could explain what we're doing. So our general conclusion right now, is that the preload dependent myosin kinetics really modify the relaxation rate, the mechanical control of relaxation, and that it's through myosin. We're not sure yet which states exactly are preload dependent, and we need to start working with individuals who do myosin kinetics to do this, because we're really focusing on the full muscle level within my lab. I do want to point out for those of you who are interested in myosin, the typical myosin cycle goes detach, attach, power stroke, ADP release, and then ATP attachment. You get that normal cycle. But there are models, Smith and Jeeves published one, Ken's published one in 2014, and there's work by Brad Palmer that we recently commented on that really alludes to the possibility of a myosin ADP detached from actin state. So that kind of is an odd state, but that's a really interesting state for us to be thinking about, as that's kind of, we're stretching the muscle, so maybe that transient state is actually really important in the relaxation. And I'm kind of excited about looking further into that state. But the idea here is that we've taken now a computer model, looked at real physiologic data, and have identified myosin as a key indicator. And this is kind of where we got stuck when we got hit with our shutdown orders. But I want to tell you a little bit that there's exciting places for this to go. We're, of course, looking at myosin. It's another version of the ATPase rate. And if we're excited about myosin, if anyone's excited about myosin, they've probably heard about this drug, omecamtiv mecarbil, because it's out there and everyone's using it. What it's doing is that it's modifying this ADP release rate also. We've done some preliminary studies on this, and we're hoping to get back into it soon, is that increasing the omecamtiv mecarbil actually increases the rate of relaxation. This was surprising to me, because omecamtiv mecarbil was developed as a way to prolong contraction, which will delay relaxation. But what I think this is important, because if you look at the clinical trials related to omecamtiv mecarbil, they report no meaningful changes in diastolic function. So in my world, I look at the work, for example, of Paul Janssen, who says contraction and relaxation must be coupled. Well, in this case, omecamtiv has decoupled it, because we've increased contraction but not changed relaxation. So mechanical control of relaxation might actually explain why myosin activators are not modifying diastolic dysfunction. And that might actually be really beneficial for people who have these diseases and as treatments. On a mechanical level, we started working with Tom Irving and Wei-King Ma at IIT and BioCat. And we've got some nice preliminary data, although it's very hard to work with these intact muscles sometimes, and it's even harder when you're trying to keep it within an X-ray diffraction beam. But partially confirming our computational model is we do see evidence of increased myosin detachment when we're stretching this muscle. And so we're hoping to use more of Tom's expertise to really look into what is happening with these muscles. I also am going to give you a teaser about Titan. I am not entirely down on Titan. We've got the mutant RBM20 mutant rats, which of course have a very broad size of Titan, and it changes the passive stiffness very dramatically. Well, it turns out that our preliminary data, when we are changing the preload, is that with the wild type, again, we see an increase in mechanical control of relaxation. With the homozygous, we actually don't. So as we do more of these experiments, if this holds up, this is telling us that Titan might actually be really involved in what's driving myosin. Some of you who are interested in diastolic diseases or even cardiac function in general, you might ask about collagen. We've started a project to look at cardiac muscle where we degrade collagen, and we again see an increase in mechanical control of relaxation. And we were able to do this also in individual myocytes. I had a great undergraduate who's now graduated, going to med school, do these experiments in spontaneously hypertensive rats and with Starkioto rats. And again, the hypertensive rats would have increased fibrosis, but the cells themselves don't have a change in mechanical control of relaxation. So it is the collagen in there that is really driving this. I'm going to quickly end by jumping into, I'm also interested in what's happening in the whole heart level. We have, I showed you before, principal component or principal axis strains. I'm really interested in knowing what's happening with these strains that are going along the muscle fibers. And as many of you know, these fibers have this neat basket weave. And so currently I'm working with the collaborator to look at ultrasound measurements to do this in vivo so we can really do active tracking of the strains. What I know I went a little bit fast, but what I hope I've kind of given you guys a sense of is that there is this idea of mechanical control of relaxation. And mechanically, it's not afterload, but it's the strain rate that's modifying the relaxation rate. We've got really nice data that's preload dependent, and we have some good data that's definitely or almost certainly driven by some myosin kinetics. And we've got some nice preliminary data that Titan and even the compliant attachments to collagen might be driving this. And we're really excited to move forward on these studies and look what's there. I do think that there's a lot of clinical relevance to this. I think that we can modify this in vivo, and I think that these strains might be a nice way to look at diastolic dysfunction. With that, I need to thank everyone, current and former members of the lab. These are all collaborators that have been actively collaborating on different parts of these projects, from Ken, who helped me develop things, to my clinical collaborators. And of course, I must say thank you to Davor, Michael, and the ISHR for giving me this opportunity and letting me close it out for you all. So thank you for that, and I look forward to answering the questions that I saw pop up but haven't read yet.
Really great talk, Charles. Really appreciate that and found it very interesting. And so I was reading through these questions as they came in. There's a couple really good ones. And so I'll go ahead and read them off for you. A couple of them were ones that actually I had as well as they came up. So first here is Alicia Mariazzi says, hi, Charles, it's good to see you. It's not really a question, but she's just, but her question here was, when you're referring to your systolic stretch, this proto-diastolic period that you mentioned,
yeah.
what is what is actually causing that aspect? She says, is this some series elastic element within the sarcomere, or what's generating that protodiastolic systolic stretch?
Ooh. So first of all, Alicia, it's nice to see you too. I hope we can meet again. Alicia was in Arizona when I was there for a postdoc for a short time, and it's very nice to hear from you again. What is causing the stretch? So this is interesting, and in the clinical, in the literature, again, from I think it's really 80s and 90s literature, is that the series elastic element in the heart. This is work I think that Washington State Ken Campbell did partially. Is that the series elastic element in the heart in the in vivo system doesn't seem to modify doesn't seem to affect function. And what I'll bring that up, one as a caution for those of you who are interested in this, is that we've found that papillary muscles are really hard to work with because they have that series elastic element. But in trabecula, they don't have that. What I think is probably happening is you get a sequential deactivation of the muscle, and so the internal elements of titin and the external elements of collagen are deforming, but we don't know what that mechanism currently is. And I think part of that for me, I'm a little bit hesitant to say exactly what it is because I don't know how the heart's really moving along those fibers. But I do think that both titin and collagen, which collagen is definitely a component of that series elastic element in our models, is really driving some of that motion within a real heart. So the systolic stretch is elastic, I do believe, but what those elements are, I'm not 100% certain.
All right, another question here from Gabrielle Sciatarella asks, what is the range of preloads that you tested for which you can observe this preload dependence of mechanical control of relaxation? But I think the question is really getting at here, her follow-up, which is that do fibers from low pressure chambers, such as the RV or the LA, exhibit similar behavior? And
Yeah.
you had mentioned when you were looking at papillary muscles that maybe other muscles were different, and I thought maybe this might be getting at those differences of where the tissue comes from.
So we have to be a little careful when I say different, because the papillary muscle difference is what I just mentioned in response to Alicia's question, is that the chordae really matter. The attachment really matters. And that's a subtlety, I think, in biomechanics. And if you look at the classical trabecula experiments, they actually use a basket and not a hook. So the muscle could actually swing back and forth in a basket. Experimentally, it is much easier to get a right ventricular trabecula and papillary muscle rather than a left ventricular papillary muscle or trabecula. As far as I know, and I actually have to double check this now because this is actually a really good question. I have to double check this because we do have some muscles, some even trabecula, that did not have good mechanical control of relaxation. So if it is chamber specific, that would be a problem for me. But I think that from all of the data we've looked at so far, we have not seen a difference. We typically get right ventricular muscles just because they're easier, but the left ventricular muscles, as far as I know, are the same. I think that some of these effects, if you're interested, for example, in skeletal muscles, some of these effects are probably relevant. If you think of eccentric contractions, at the end of an eccentric contraction, it's a very similar feature to what we're doing, which is stretch. To backtrack a little bit on this question and think about what's the range of preloads, the trabecula are a little bit weird in their range. So we did a 5% length change from maximum length, which we assume to be about 2.2 or 2.3 microns. So we're getting close to slack length, and we're still getting this relationship. It is harder to go below that length, so I'm not sure if other muscles will see that. And I'm not sure if there's a threshold. There's actually some data I'm thinking from, I want to say it's the Leap Merita group, where there was like a kind of a bimodal change in their relaxation rates when they did modifications, I think like preload. So I think there's potentially a threshold, but for diseased hearts which are often hypertensive, or in a hypertensive state, for example, I think this is going to be a really interesting feature.
Cool. All right, the next question here says that assumes basically you've done all of these in crystalline buffer solutions with glucose as an energy source. Have you tried this using fatty acids? I think the question here from Dr. Anonymous is there a metabolic role in relaxation?
That's really interesting. And we use crystalline perfusates. If you looked at the subtleties of our experiments, we actually use pretty non-physiologic conditions for our retrobecs. We've been doing low pacing rates. A lot of it is at room temperature. We haven't tried it yet. I think that's really interesting. I have thought about, we haven't had the people or the time to do it, about trying to modify mitochondrial function and really seeing if that really could modify this also. And that would be both changing calcium and myosin because of the ATP availability. So I think that's a really good idea. We haven't tried it yet, but it's something that I think is a really a good future direction for this type of work. Good to see Mike's comment. Good to see you too, Mike. I'm glad you joined in.
All right, so next question from Laura Sommerfeld is regarding the speckle tracking that you did in vivo. Can you also look at the mouse right ventricle using echocardiography data?
I don't. We're just getting in on that, and we're working on now really driving the in vivo function of that. I think we're going to struggle with the right ventricle. I've worked with some individuals who've done MRI. If you use the 7 Tesla MRI to do mouse work, you get maybe 3 pixels through it. For our echo, I'm really worried. I don't think we're going to get great resolution on that, unfortunately. I would love to, but I would say let's be hopeful, but don't expect that anytime soon. But I think it's a really good thing, and the right ventricle's got a very different weave, and that affects the function. Now, when we get to pulmonary hypertension, that might actually be something we can look at, because we do get that hypertrophy.
Jonathan, could I just chip in and ask a question, if I may?
Yes, of course, please.
Charles, to what extent does any of your mechanical interventions influence the calcium transient? And the reason I ask this is that I've had a PhD student just working on the relationship between QT interval and diastolic function. And QT interval is just linearly related to the calcium transient, but doesn't affect diastolic dysfunction, particularly in the models we've got of HFpEF. So there seems to be this dissociation between what the calcium transient's doing and what diastolic dysfunction. What's the relationship between calcium transient, mechanical control of relaxation, and what's more important, the shape of the calcium transient or these mechanical factors? Can you just sort of expand on that a little bit for me? This is just showing my ignorance, but it'll really help.
I think that's a great point. And when I got into this, I found I got to look at this paper again. But when I got into relaxation, someone had published that in an isometric trabecula, the relaxation rate was directly related to the calcium uptake rate. So they basically took those 2 taus and they were directly related. The work from Paul Jansen's lab about contraction relaxation coupling really points to you can make tons of modifications to calcium, and the major thing that's modifying relaxation is really the sarcomere kinetics. Now I'm a sarcomere function person, so we could argue, and there may be some good reason why the calcium is important. But like you said, modifying the calcium transient doesn't seem to do a whole lot to diastolic function. And I think that's because the calcium transient has dropped by, the calcium concentration has dropped by so much by that point. The other really interesting thing, and that we haven't really gotten as deep into this as I wanted to, is that first part of what is the mechanical perturbation doing to calcium. This is hard, and there's really varying evidence in the literature about what happens to the calcium concentration. For example, even when you change preload. I'm reasonably confident now that there's enough data out there that when you change preload, the magnitude, at least, of the calcium transient changes. The question for me is, does the shape change? The other part is we had some hints of this, but we haven't done enough calcium transients to know. When we stretch the muscle, there may actually be an increase in Fura-detected calcium. And the problem is we don't know if that's just because calcium is being released from the thin filament on that stretch, or if it's actually releasing calcium from the SR when we're stretching it. So that's a really interesting question, looking at what's happening with calcium. As far as I've looked at my models, there's a very limited effect on if I change like the calcium reuptake. If I change calcium reuptake, it typically just prolongs contraction a little bit. But if I remember right, mechanical control of relaxation is pretty much constant.
I guess, Charles, the thing that we found, because we cite that paper on isometric contraction as well, but when we're actually looking in working hearts, and we had a working guinea pig heart, which has got a normal action potential and, auxytonic contraction, etc. When we looked in that, we couldn't see this relationship between calcium transient duration and diastolic dysfunction. So we thought, okay, well, we'll screw up the SR. We'll slow everything down. We'll give cyclopentazonic acid. We'll screw up sodium calcium exchange. And we screwed all these things up. And we even then looked in a real model of HFpEF. And everything we threw at this thing, the calcium transient didn't seem to really influence diastolic function. And that was a bit disappointing for my PhD student, who whole PhD was based on the idea that it would. But the reality that we found was that the calcium transient, even when we prolong it and mess around with it, as we know happens in heart failure, really that still didn't, and even at high heart rates, that still didn't impact on diastolic function. So I'm coming around to the idea that I should be paying attention to what you are doing, because you're telling me that these processes may be more important in determining relaxation rate, diastolic dysfunction, etc. So it's good to hear that from you, somebody who knows what they're talking about, as opposed to me.
I appreciate that. And really, I mean, again, this is kind of influence on my background of the heart springing back. Yeah. And it can't really spring back without those contractile processes having already been down. And if you even look at the Brute-Serret's early work, if you modify the stretch before peak force, that's not what's modifying relaxation. It's after the peak force starts coming down, which means the thin filament must have already started to deactivate. I think the level it comes down to might matter, but I'm not sure if the timing, because I think if you mess up the timing of calcium so much, where you drive basically the reuptake to be so slow that you would increase the diastolic level, then you might get some changes in filling. But really, a lot of that's going to be early filling, where that early relaxation delay is going to be impacting the system.
So actually, that's a good segue into this question here. Sorry, I'm just going to pop in here with this question because somebody was actually asking. Brute-Serret proposed actually that the cardiac endothelium plays a role in modulating relaxation, and I love me some cardiomyocytes, but is there a role here for other cell types to be involved?
Um, I wonder if that relates a little bit to Alicia's question of what are the elements, because the endothelium of the ventricular endothelium, I wonder if that's what they're asking about.
Yeah, yes. The layer of the endocardial endothelial cells, I
Yeah,
believe. Yeah.
I would say in conjunction with the like the collagen and elastin, which are affecting those elastic elements. That's a very important thing. Um, whether or not the endothelium is driving it, that's an interesting question. So all of our trabecula have an endothelial layer on them. Our collagenase experiment might be modifying
I
that.
think, Charles, I think Broussard had even gave it a name, didn't he? They had endocardin or something, which was something that was released. It was a paracrine secretion from the endocardial
Yeah.
cells modifying relaxation. And when you triton stripped off the endocardium, you ended up with a different relaxation pattern. I think that's the story. I don't know whether anything came of that.
Yeah, I'll have to relook at that because I think that our collagenase experiment might be similar to that. And really, I'm wondering if I can differentiate this endothelium response with collagenase digestion of those cells, because we could be killing the cells that are releasing this endothelium thing. So it's a very interesting point. I'll have to go back into that aspect of his work. I think the other related part of Broussard's work, when he talked about triple control of relaxation, he talked about afterload, which I think is strain rate, but he also talked about the asynchrony necessity of the muscle. So if one part of the muscle relaxes before the other part, and I do think that that's a mechanical aspect that we are working with that is important. So we'll hopefully go back into looking at, for example, videos of this to see what's happening to those muscles. But yeah, I highly encourage anyone who's interested in relaxation to go look back in Broussard's stuff, because it's, when they say it's been done before, he did a lot of this stuff before. We're primarily just interpreting it a little bit differently. And do go back and look at Weir's original work too, because it's kind of amazing to look back at some of those old studies.
Jonathan, do you mind if I just follow up on actually what Mike has asked, as well as, I guess, to some extent, this question? I mean, Charles, you showed in your talk the effects sort of extracellular matrix due to fibrosis, and I actually, I mean, I'm sort of working on a project of cardiomyocytes, cardiofibroblasts crosstalk, and I'm beginning to kind of ask a question of what is the stiffness of the myocardium in sort of healthy and fibrotic hearts, and I'm struggling to find a sort of a real answer to it. But my question here is, and it's in relation to Mike's HFpEF stuff as well, but also in relation to other cell types. What's stiffer? Is it a fibrotic heart, with maybe interstitial fibrosis, such as in a HFpEF model you might get, or the raised diastolic calcium level? What you think is providing more of us kind of that stiffer environment that's preventing relaxation? Or do you think both are contributing equally?
I think both are contributing. It's a question of how much. So I'd say when you have, for example, diffuse fibrosis, or when you have prolonged hypertension, for example, and you get fibrosis, I think that's definitely affecting this mechanical feature. And one of the things it actually does is it prevents the ability of the muscle to not only lengthen, but actually shorten. So there's actually a link between how much it shortens and lengthens. I have not been able to get too deeply into the diastolic calcium level. I think it matters, but I'm not sure what that's doing to stiffness. There's also a little bit of the diastolic calcium has more of a viscoelastic effect. So depending on the speed we stretch our muscles, that's going to modify this. So I think that's an interesting point. it's something I think we need to keep going on. And it's the idea for me is right now I'm focusing on stretches that are really early in the cycle, not late when for example, we'd be looking at filling. When we're looking at filling, my bias is that the elastic is going to do more than the diastolic, but I think the diastolic really matters. And there's work that I think it was the group in Amsterdam at the VA. First author was Vasco Sequoia. I'm blanking on the last author's name, and I'm really embarrassed because I've worked with her before. But I think that diastolic calcium really matters. To what extent, I can't give you an answer, unfortunately. Maybe in a couple of years. We'll do this again in a couple of years. We can.
I had a couple more questions. So we have one here. Says, do you observe the preload dependence mechanical control of muscle contraction and changes in contraction kinetics?
Yeah, so we're getting a little bit more into that too, I think. And I think this is Danuta. I think what you're asking is that when you change the preload, you actually change the kinetics of force generation. So for example, if we did a KTR maneuver in a skinned muscle at long versus short length, you change the states. And this is, I'm sort of using the computational modeling as a surrogate for these changes in contraction kinetics. We haven't had a lot of good experiments yet where we've been able to do that directly. I've been looking into, I think I keep mentioning Paul Jansen's work, but we've been looking into his work on trabecula, where you can actually get KTRs in living trabecula and have them pace again. So I hope we can answer that question soon. But right now, all of my hints on that are really within the computational modeling, showing that it does change, and also a little bit in the experiments that we did with, for example, omecamtiv, where we're seeing some changes.
Next question is from Chris Solis, and he points out, of course, that in vivo, muscle is not just exposed to a single axis of strain, and the muscle is also being exposed to lateral strains as well. And so could these multiaxial strains on the muscle actually affect the mechanical control of relaxation as well?
That's interesting, and I've thought about ways of trying to decouple that. I've also thought about ways of pushing on a muscle while it relaxes. The problem with pushing on a muscle in my in vitro systems, you can imagine if I've got a muscle here, if I'm pushing on it, I'm actually straining the muscle too. I think that I would have trouble saying that it is. I think if it matters, part of what matters is, as you say, we've got this weave of the heart. So if I'm straining this aspect right here, then it's gonna also strain the next aspect just because they're coupled. I think that's my answer right for now. And let's see where the science takes us. If we can get a good way of measuring that, I'll be excited. I'm a little bit worried about that.
Yeah, that sounds pretty tough. And like you said, I mean, it's sort of a theme throughout here with relaxation is all these things are coupled. And so teasing apart the individual effects seems challenging.
Yeah, and I think that's what makes relaxation actually really hard, and why we have really found those issues.
So we've got a follow-up question here on the one that you answered earlier about the kinetics. How much changing the contraction kinetics would affect both mechanical control of mechanical control of relaxation, and can you uncouple those?
That's a good question. I'm not sure we can. So this is where the omecamtiv experiment comes in to say, I'm changing, if I'm changing that kinetics. What I'm excited about doing right now is I'm really looking into the different, I'm hoping to take this computer model a little bit deeper and then work with someone who can do myosin kinetics and see if we can identify the different stages where that gets modified. And I'm really interested in this strain-dependent detachment. I don't think that changing that would dramatically change the contractile kinetics, but it would change the strain rate kinetics. And so that's where I think it would be really able to uncouple both. But if we're really just talking about the power stroke, or if we're just talking about the rate of detachment or attachment, I don't think you can uncouple it. I think that they're uniquely coupled there.
All right, Kathleen Wolf has a great question here. Are there any sex or age differences in the slope of the mechanical control of relaxation?
So I will say we haven't tested that yet. What's interesting is that we kind of historically kind of just used female rats for a long time. Another caution that brings up is that we haven't checked estrus when we dissect our rats. So we know that it exists. We've done it in male rats. I don't think we've fully characterized that yet. And we're waiting on hopefully a grant under review right now that will let us do this experiment, because that's one of the proposals in there. In terms of the basic function, I don't think it will be different, but in terms of the in vivo responses, I'm not sure. And I think that, and I'm looking at Casey's follow-up question about species difference, and I'm going to lead into that in a second. I think that one of the differences is that there may be some protein differences that are affecting this. So for example, if we affect myosin or one of the troponin isoforms, maybe that modifies this. And then I'm seeing Casey followed up with what are the species, there were species differences,
mm-hmm.
and could there be differences? So that's really interesting. And when we published that paper, I was interested in it, but I couldn't really get anything out of it. The mouse had a higher slope than the rat, which had a higher slope than the human. I think that that's a mix of myosin and titin. I think that the myosin isoforms are modifying this. So the more alpha, probably the more sensitive, and I think also the stiffer titin, the more sensitive. We're interested in that. And I think that our current experiments, we've got some experiments in rats where we're modifying the myosin isoform. And then again, we've got those titin mutants that we're working on. And I think both of those will go a long way into answering that question that you just asked. So knock on wood, I'll have enough funding and personnel to work on this. And we'll kind of hopefully answer that question in the near future.
I have a quick question too for you on is there any role here for the super relaxed state of myosin? Are these just heads that are out of the picture and so they don't play a role, or is there something that they're contributing?
So I glossed over the modeling part of that, and the modeling part of that is that the super relaxed state does not modify mechanical control of relaxation. And we actually have done some experiments with the mavacamten, the drug that's supposed to drive this effect. Again, we don't see a change in mechanical control of relaxation with this. And I think that the reason for that, I'll caution that, is when we go to super high doses where we get very low forces, we start to see a change. The caution I'll give on this is that the super relaxed state means that, as far as I understand it, means that the myosin head is not cycling. So since we are looking at detaching a bound head, that super relaxed state is actually not going to be part of this situation. And I think a lot of this depends on how it drives in and out of the super relaxed state. So if it can drive in and out very quickly, maybe it won't matter, but I don't think that the super relaxed state actually drives this.
All right. So seeing no more questions for Charles here, I will say thank you very much, Charles. I think it's been a stressful couple of months, and so we could all use a little more relaxation. And I think this is a great place for us to end.
Thank you
And
for that. Thank you for, as I said online earlier, being a better than moderate moderator.
appreciate that.
Thank you, Gavin.
Thank you to all the attendees who continued through to the very end. We really appreciate it, as well as the organizers who kept this going with all your hard work. Like I said, it's been something for us to cling to in a sort of a normalcy and to continue to make interesting conversation and progress in our thinking. So thank you again, everyone.
Thank you, guys.
Thanks,
Bye-bye.
Thank you.
See you in September, hopefully.
To be continued.
Bye-bye.
Bye.